Well, you mentioned prostate cancer there.
Right.
I want to bring cancer in as a whole.
Okay.
And in your book, you share a couple of studies that were linking glycine to cancer,
Right.
or at least inferring that.
Right.
Talk about what those studies were showing,
Mm-hmm.
and then your thoughts on any connection there.
Oh, okay. Right. Well, there was a study that came out in 2009. The head of the group is Arul Chinayan at the University of Michigan. And they published a study that showed prostate cancer in cultured cells. They said that glycine, they had done this introduced in my book, a discussion on what we call metabolomics, the omics disciplines, where they can study a gazillion things at the same time. So they were doing a metabolomic study, looking at the various metabolites, metabolic intermediates, all the biochemicals involved in that were in blood when you had like prostate cancer or normal. They were looking at prostate cancer. They were looking at cultured cells in the cell medium in prostate cancer. And they found that prostate cancer, that glycine seemed to be elevated. And if they then tried experimentally, and if they added glycine to the medium, the cancer, they used a model called RWPE cells. They're cells. I got to give a little background on tissue culture. Tissue culture, where they grow cells in the test tube, right, in the petri dish, and they passage them for generations and generations. So if you're a cell biologist studying these things, your grandson or your granddaughter can be a cell biologist in another 40, 50 years and study the same cell line. You keep passaging them. Well, normal cells you can't passage indefinitely. There's something called a Hayflick limit. So you can't passage cells forever. You can go a few generations and then they die out. So there is one line of cells that were derived from a prostate patient called RWPE. The RW is probably the patient's initials, and the PE is prostate epithelium. And these cells are immortalized. So they use them now in prostate cancer research. When they do cultured cells, they use these RWPE cells as controls as if they are normal. They're not normal, but they act like normal under most circumstances. And they are immortalized so that you can passage them forever like you can cancer cells. So they found that if you added glycine and sarcosine, which is a metabolite of glycine, you can get these quiescent cells to become invasive and malignant in their behavior. So this was an interesting finding. And of course, these are, first of all, not normal cells. But what's also interesting is that 5 years later, very quietly, that finding was retracted. They didn't quite retract the finding. They published another paper, a follow-up paper, where they said, right in the bottom line of the abstract, they said that sarcosine, but not glycine, induces a malignant transformation in these cells. So that one finding that raised the alarm turned out not to be reproducible. Because again, the problem is you're using cells that are not normal as normal controls, just sort of one of the natural or the normal weaknesses of doing any kind of experiments in tissue culture, because you can't use normal cells for long-term tissue culture. So anyway, that kind of went by the wayside. Then there was another study on breast cancer, which showed that most breast cancer cells will actually utilize glycine and to grow more invasively if you give them artificially glycine supplement. Well, so that was interesting. It caused me to kind of look at the whole situation from a point of view of cancer evolution, if you will. We all know cancer or abnormal cells, potentially cancerous cells, happen. They develop all the time in a normal body, but they are taken care of by something called immune surveillance. The immune system spots them and takes care of them, usually knocks them out. It's a complex interaction between lymphocytes and macrophages of different kinds, not fully understood, but at least in broad strokes, it's kind of understood. Well, when a cancer is developing in a body that has chemical conditions that are conducive to development of cancer, diabetes and other conditions which we know predispose toward cancer, not to mention having certain genes that do so. When that happens, there's a selection process that goes on with each generation of cells that are on the way to becoming cancerous. And so as they become cancerous, the more malignant a tumor gets, the more efficient it gets at just multiplying and spreading in the body and not doing the ordinary differentiated functions of that cell type. So they become de-differentiated. So one of the things that ordinary cells do is they have to have this, remember I talked about the whole methionine cycle, those methods of regenerating and recycling methionine. Well, if you're in a body that's overfed and got lots of methionine and is deficient in glycine, you're going to end up with cancer cells that are deficient in recycling methionine because they don't need to. There's always plenty around. And you will find that not only are most human cancer cells more sensitive to methionine deficiency, but there is even a treatment modality. There's a practitioner, his name is Bob Hoffman out in California, who treats patients with something called methionine-ase. It's an enzyme that actually gets methionine out of the blood plasma to stop or help to stop the cancer from growing and spreading. I'm not sure how successful it is, but he's been in business doing that for some years. So it is certainly known that those cells are not good at recycling methionine because they evolve in a body that doesn't, where they don't need to because they get lots of methionine. In the same way, those bodies tend to be glycine deficient. So what do you get? The successful cancers are ones that are more efficient at utilizing glycine. So you give them glycine, they'll utilize it more avidly, more readily, and more efficiently than cells that are normal cells. So you might say, well, isn't that bad? Because if you're going to add glycine and there's cancer around, isn't that going to make the cancer worse? Well, if you look at the experimental protocols, when they grow these cells to show the evidence that these cells grow more quickly, more efficiently, if you give them glycine, how much glycine do you give them? Well, the control cells, guess what? They don't give them any glycine. The cells that they're treating with glycine, they give them about 100 millimolar glycine. Oh, look, we give them 100 millimolar glycine. Well, guess what? You're going to get more than 100 millimolar glycine in anybody's plasma. So you're never going to get glycine low enough to stop these cells from growing. So trying to control the glycine level, it's a great way to control inflammation because you just raise it. But if you want to lower it to some level where it's really, really low, you can't do that. Glycine is the most abundant amino acid in the body. You need it for so many different proteins, for collagen, and for everything else. You can't possibly get the glycine low enough that you're going to make any difference in a cancer cell's ability to utilize it, because they're going to be able to utilize a small amount of glycine better than a normal cell will. So that's kind of my bottom line on those things. I can understand now, and that was important to me just scientifically. Why is it that not all, but some plurality or even majority of cells are bad at recycling methionine, and why are they particularly good and better than normal cells at utilizing glycine? It makes sense from the point of cancer cell evolution in the body. You're going to get cells that are evolving in the body that are rich in methionine and poor in glycine. Guess what? They're going to be able to use glycine more efficiently, so they can use lower levels more efficiently, and they're going to not be able to recycle methionine because there's always plenty around. So it makes sense from a cancer evolution point of view. So all of these, as I say, kind of every line of evidence. I know you're trying to think of some others that maybe I haven't thought of, and that would be a very good thing. Maybe we'll discover something new on this podcast that's of use to everybody. Dialogue produces a lot of good discoveries anyway. It's a great thing. But anyway, that's where I am, and that's sort of what I cover in the book.
So in your mind, is there any diseases, chronic health conditions that glycine could possibly exacerbate or
Oh
lead to?
yeah, right. Well, there's what's called NKH, non-ketotic hyperglycinemia, which is a disease in which glycine synthesis is uncontrolled. And so there's too much glycine around, and it is characterized by someone who appears to be perfectly normal at birth. And then as they age into a few months of age, they progressively start getting more and more seizures. And usually they die at a very young age. Now, I guess they're, gradually, incrementally extending their life. They use things like adding benzoate to which binds glycine to try and excrete it as fast as possible. And that seems to be has to do with the nervous system, that glycine as a neurotransmitter. It's actually a neurotransmitter of two kinds. It's an inhibitory neurotransmitter on its own with the classical glycine receptor. And there's also a receptor called the NMDA receptor, which is a so-called a GABAergic receptor, a more activating receptor, where glycine is a co-activator. It's one of the two activators of that neurotransmitter that enables ionic flux, I'm not sure what kind, that transmits a nerve signal. And those neurotransmitters, those neurons seem to be very, very sensitive to too much glycine. And so, you certainly don't want to give people glycine like that. You want to get rid of it as fast as you can. And it seems to be that NMDA receptor that's the culprit. Now, I saw one thing in the literature, and I suggested to one practitioner who was treating kids who have this condition that he might look into DHEA, dehydroepiandrosterone, which is an adrenal steroid that because it seems to it was shown in one animal study paper that it inhibited the it seemed to protect the NMD receptor from glycine toxicity. The reason that makes sense to me is that in utero, your DHEA levels are ordinarily very, very high. The feto-placental unit, as it's called, produces enormous amounts of DHEA. And then when you're born, and the tube is tied and you no longer have a placenta, the production goes down to zero. And you don't make any DHEA until what's called adrenarche when you're about 8 years old. And then your adrenal glands start producing more of it. So I just suggested that, and I never really got a reason why they couldn't try it, except, clinical trial constraints and so forth. And it's not really my field. It's just a suggestion that I passed off to one clinician. But that's the only one that I know of where you have a genetic condition like that. I think it's a recessive condition. If it is, it might be interesting to look at conditions where there is ordinarily too much glycine, but only with one chromosome, so it's not enough to be toxic. Does that make people live longer? Well, there is a study from the Cleveland Clinic from, I think, 2015. This is about a genetic condition where the glycine level affects the health outcomes. So this group did a study on a genetic condition called CPS, which is carbamoyl phosphate synthase type 1 deficiency. What is that? Well, carbamoyl phosphate is the first step in the urea cycle, where the kidneys mainly take ammonia and turn it into urea, right? Not efficient, not good to excrete ammonia, so it's toxic at high levels, so it's turned into urea. And so there is a cycle for it, a urea cycle that it feeds into. And the main substrate, the most abundant substrate for this is glycine. It's the most abundant way that glycine is actually cleared from the system is through donating its amino group, like that's the ammonia source, for making urea, which is a nice, safe, water-soluble compound that we can then excrete in the urine. Well, so the Cleveland Clinic, of course, is a very big place. So they got a lot of data from a lot of people. And they looked at a database with something like 67,000 people. And they came up with something like 1,200 people with a genetic defect. And they had people who were heterozygous for CPS1 deficiency. Well, I say heterozygous, which means they had one gene for it. Why? If you have two genes for that, it's fatal. You got to be able to make urea. But you can make it with one gene. However, something very interesting happened. This was a metabolomic study. And I think metabolomic studies, where you can measure lots of things at the same time, it's kind of like AI. It can do so many things, it actually dumbs down the field. You get less kind of independent, real intelligence working. But and people use it all the time in so-called data-based science. They run these metabolomic studies or genomic studies in order to generate hypotheses. I think they're good at testing hypotheses, because you generate a hypothesis and then you have a finding and you don't know why, then you have to think of why. So this is one that linked up with what was already in the literature. And typically people who do these studies, they don't even link it up with what's already in the literature. But I'll get to that momentarily in terms of the scientists studying it, the doctors who were studying it. So they found something like 1,200 patients. And what they were looking at was, these were the records of the people who were treated at the Cleveland Clinic for what? Cardiovascular disease. So what they were looking at was the incidence of MACE, major adverse cardiac events, basically heart attacks and strokes, right? So they looked at the incidence of MACE, and they found that women didn't hold for men, but for women, probably because men and women's kidneys are different, just like men and women's livers are different, because the sex hormones cause changes in their activity. And they couldn't figure out why. But for women, they found that the CPS deficiency, having one bad gene, significantly lowered their risk of having a heart attack or stroke. It was something like a 12% decrease in the risk of heart attack or stroke. Very, very, very significant, like P less than 0.0001, because they had so many patients. And they looked at so then they did a metabolomic study. They measured, a gazillion metabolites, a few hundred actually. And they found that glycine stuck out. The computer said, glycine is high in these women. So they said, why is glycine high? Well, because glycine kind of backs up a little, waiting on line to get into the urea cycle, because there's only one gate instead of two gates. One enzyme, carbonoyl phosphate synthase, not two. So the glycine kind of backs up a little, and the steady state concentration of glycine in the plasma is higher than normal. Not higher than natural necessarily, but higher than normal. So they did a very good statistical analysis to show that if they cleared away any other reason that this might be happening, nope, it's the glycine. And so they said, no, it's glycine. We don't know quite why glycine is protective, but it sure looks like glycine is protective. And again, it's one of those things where I say they didn't know what was in the literature. They should have known that the Thurman group, just a couple of years prior to that, had identified the glycine receptor on, guess what? Platelets. Glycine actually is the body's natural blood thinner. It inhibits platelet aggregation. Again, not to a pathological level. You don't bleed to death like you might even with aspirin. But so they didn't even say that in the paper. They had no suggestion it was platelets. They threw around a bunch of hypotheses. Maybe it's this, maybe it's that. Because remember, it was a data-based study where they started with the data and then tried to figure out what was going on. But clearly what's going on is that certainly women who have, probably the men don't have higher glycine levels, but the women had higher glycine levels because they had a defect in this enzyme, which uses glycine as a substrate. The glycine is backing up. They had higher glycine in their circulation. They had a significantly lower incidence of heart attacks and strokes. So that's one. I mean, we could go on all day, I guess, but you may have gotten to all the ones that I know of in terms of disease states that are affected by glycine. And I don't even know if I covered them all in my book, but this is one of the things that's there in the literature. And which also makes me a little sad because, you'd think when somebody discovers something, and here was a new finding, a discovery, an important discovery, it never gets, it just never gets tied into what's already been discovered and reported. And this is what my hope is, that, if AI really gets some of the bugs worked out of it, I would love to be able to tell an AI robot, okay, read every scientific paper ever published. Should be easy for AI, right? And tell me, what is it that, that actually cures cancer or stops heart disease? And I'm sure it would come up with this one, because if I could come up with it, it's in the literature. You can just do it. So a lot of research is asking the right questions. And when it comes to the literature search, it's basically using the right search terms, which is really a way of asking questions to see what will come up. But you have to start with some idea of what you're looking for. I don't really believe in fishing expeditions, which is what all this database science tends to be about. Generates an awful lot of data and a lot of, half-baked hypotheses that just kind of lead people on rabbit trails that don't go anywhere. I mean, so this was the whole hypothesis about glycine. It's one of those things. I have a gazillion hypotheses a day, as you could figure out, probably, right? I mean, as a scientist, we're generating hypotheses all the time. And, when you have one that has some significance, you hold on to it and you test it. You test it this way and you test it that way. And you see who else in the literature has tested it this way and that way and the other way. And, and eventually it just, it's like recrystallizing something and purifying it. And that's what comes out to the whole hypothesis about glycine and inflammation. And I just believe that it's one of those things that, there's just not good evidence that inflammation in the ordinary levels that we experience it does any good at all. I've even talked to a couple of people. Oh, I don't need your product. I do bone broth. I have bone broth every day. I love it. I said, well, do you ever go out in the sun? Oh, yeah. You ever get sunburned? Nope. Fair-skinned as anything. You ever get a sunburn? I get a little red sometime, but otherwise, no. They don't experience the kinds of things that some of us normal diet people would otherwise need a glycine supplement to normalize. And as you point out, if the diet were really healthy in the first place, we'd be in that kind of situation.
If you enjoyed that clip, you're going to want to head over here and catch a full episode. I'll see you over there.
Contrary to what it says in any source you look at, that inflammation is the immune system's first response to infection or injury, it is really only a first response to infection. Inflammation does the body no good.